I guess it will have it here.
Think it's working.
Okay.
All right, then we can start. Okay, so I'd like to say good morning to all the people in the US, good afternoon, good evening to everyone else. So I'm Melanie Dammhahn and I'll be hosting today. So just before I introduce today's speaker, last week we didn't have any fine because it was the Thanksgiving holidays in the US. But if you want to catch up on the previous fine speakers that we've had, you are welcome to visit our YouTube page and you can find all the past speakers. Their talks will be are always uploaded. And the last fine we'll have will be next week, where we'll hear from Manjari Jain. And again, this will always announce this ahead of time. And you can also visit our, what's it call it? Not Twitter, but our X account. And we'll try to always post about who's coming up. But this week we have Melanie Dammhahn from the University of Münster in Germany, and she will be talking to us today based on her favorite topics, mostly because she's a behavioral animal ecologist who's interested in the ecological consequences of individual behavioral differences, so mostly personality in animals. And she did her PhD at the University of, what do you call it? University of Tübingen in Germany. And currently she's a professor at the University of Münster for behavioral biology. But I don't want to talk too much about her. I will let her do most of the talking. So if Melanie, it's possible for you to share your screen and we can get started.
So I hope you can all see my screen.
Yes, it looks good. Yeah.
I just have to move you guys away a bit. So, yeah, thanks Lindelani for this nice introduction. Also, thanks to the whole team for this invitation to talk at this fine series indeed. Now it's your chance to run away because my talk will be very, very ecological. So there's not so much about sociality or the evolution of sociality in that talk. There are here and there a couple of teasers where this could lead to also into studying variation in sociality, which I did, for example, in my PhD. But in the last couple of years, I really became very much interested in joining my different fields of research, which were, classical behavior ecology and animal ecology. And one area where we do this very intensively is that we got really interested in understanding whether there is actually individual variation in landscapes of fear, and I'll explain in a second what this is, and what is this actually good for. And to get us going, let's go back to some ecological textbook knowledge. And I think everyone who opens a behavior ecology book or an animal ecology book at some point will read about this idea that obviously one of the most important problems that animals have to solve every day is to eat, but not become food for others at the same time. And this bank wall here, this example bank wall sitting on a branch, chewing at these wonderful juicy blackberries, really has to balance this risk out. And doing so has obviously led to a lot of very important adaptations in behavior, like anti-predator behavior, and also obviously variation in sociality. And why is this so important? It is basically because predators obviously can be everywhere at any time. They are often very hard to predict for prey organisms. And of course, they do change prey fitness very directly. And this kind of intrinsic risk of becoming food to a predator has led to, for example, this whole co-evolutionary arms race between predator and prey. But also kind of the mere presence of a predator has led to a lot of very interesting adaptations. So simply the threat of being eaten, so let's say the perceived risk of being eaten by this predator has led to a lot of really interesting adaptations. And disentangling these two direct and indirect risks has been done really nicely many years ago in a review article in Tree, where people pointed out that there is this kind of ecological effect of direct predation, which obviously acts directly on the survival component of fitness. But there is this indirect loop via anti-predator responses, which then lead to energetic trade-offs, often energetic consequences or physiological consequences like stress responses, and both of them then act on the two components of fitness, so survival and reproduction. And what is very important here is that this whole indirect effect loop acts via perceived predation risk. And how this is kind of mediated is basically behavior. So these are behavioral reactions. So here again, kind of the ecological scheme, you have on the left side these numerical effects of a predator directly killing its prey, and this then can have cascading effects to the next party, let's say the food of the prey organisms. Via density effects or density mediated effects. But this whole right side of the scheme, these indirect effects are acting via the risk perceived by the prey. And this is the perceived risk. As I say, doesn't have to do necessarily be totally congruent to the real risk, to the intrinsic risk. And then there's a whole set of really interesting trait-mediated indirect effects, which are all playing out via behavior. And that's why I think it's so interesting for us people studying behavior. And in a nice meta-analysis, which is also again like 20 years old, Preiser and colleagues pointed out that these indirect effects, so everything on the right side of the scheme, the effect sizes are 85% of the effect sizes of this direct killing of prey. So it is an important effect. It is basically as important as direct predation on prey. And I think that's why it's also worth looking at it and trying to understanding it a bit better. And if we think about how this perceived predation risk varies in space and time, then we couldn't depict it like in this nice modeling paper where I just steal the figure, that obviously the intrinsic risk of being eaten in a landscape is not equal, but it has these different peaks where predators like to hang around. But this kind of real risk translates into a landscape of perceived risk that can be very different. And how this landscape looks like has very much to do with the sensory modalities that the prey species is mainly using. If you get your cues mainly from olfactory signals, then obviously this landscape might be less caused than if you get it, for example, from acoustical signals. Your predator is calling and you directly receive the risk right here at this very moment. And basically this kind of spatial but also the temporal distribution of perceived predation risk is what we call a landscape of fear. Well, that's how it's called in ecology. Whether this is a good term, we can later argue in the discussion, but this is how it's called. And obviously, we need to know how we can actually quantify this fear in space and time. And there are a couple of really nice studies where people tried this, and one really nice textbook example is done by Eric Willems and Ross Hill, using vervet monkey alarm calls that they mapped in space. And these monkeys give referential alarm calls for different types of prey. So in the left corner, you see the map for the eagles, so where they perceived highest risk of being eaten by an eagle. And then, for example, in the upper right-hand corner, you see the risk distribution for being eaten by a baboon. And these different, spatial depictions are really nice examples of landscapes of risk, how they were perceived by the vervet monkeys. This is nice, but it doesn't really give us yet, let's say, a good currency to translate this into indirect fitness effects. And one method that actually allows this has been invented already in the 80s by Joel Brown, and is a super simple method that a lot of people teach in their student courses, because the idea is that you use a food batch where you have a food items in this food batch that can be harvested. And the longer an animal stays in that patch and harvests the food, the less it gets per time, because these patches are exploitable. So if you, for example, think about an animal at the beginning of this harvest curve, staying a certain amount of time in a patch, then it can actually harvest quite a bit of resources in this given time. But if it comes a little bit later, when the patch already has been exploited, then it stays for the same amount of time that it stays in the patch, it gets much less. And the idea that Joel Brown brought up is that if everything else is equal, then this harvest rate when an animal actually stops foraging in a patch should be equivalent to its perceived predation risk. And equivalent here really means equivalent in energetic terms. And this kind of translation into energetic terms really allows you to use this kind of giving up density to calculate in a currency that we use a lot in behavior ecology, how risky a given animal perceives to forage in this given place at a given time. And because this is such a handy and easy method, people have used it a lot. It comes with a couple of problems, which we also might discuss later. But this is a way to, let's say, map this landscape of fear. And again, this has been done a lot on the population level. And in whole ecology, actually, the landscape of fear is a population level concept. But I guess everyone in the room, in our virtual room, is aware that in the last couple of 2-3 decades, there's been a lot of interest among individual variation in behavior. And this is so common that we find it in basically every organism that we study. And then we just have to be smart enough basically to come up with some really good measures to show it, and have tests that are, ecologically relevant. But we'll talk about this in a second a bit more. But just for those who haven't come across, I mean, what we basically talk about is that between individual differences in behavior expressions are kind of stable over time or context. So if you think this red line is one organism, one individual, this one will always respond a little bit faster to a given environmental stimulus than, for example, the yellow one. And this does not mean that none of them is changing in their behavior. They do change in their behavior in response to environmental conditions, but they differ on average in what they do. And not everyone is basically showing the whole flexibility or the whole behavior variation that is present in the population, like depicted in this gray arrow here. And in this whole field of animal personality or temperament, or among individual differences in behavior, whatever we want to call it, there always have been a lot of discussion that variation among these individuals should basically be maintained via individual solutions to risk reward or safety reward trade-offs. And so if you think a little bit about this whole area and try to join them both, then obviously the question comes up: is there something like individual variation in perceived risk? And if so, does it actually matter? Do we need to think about it when we think about ecological interactions as the one via like indirect predation effects? And this is exactly what I asked myself and what I want to talk about today. And very specifically, we asked kind of 3 big questions. One is, does actually perceived predation risk vary among individuals? And if so, then we would expect that foraging under risk, like in these rabbit monkeys or using giving up densities, is actually repeatable over time and across contexts of risk. So the classical definition of animal personality. Then another question that I ask myself is actually this risk-taking during foraging and boldness, so this classical animal personality dimension, are they functionally integrated? Is it the same thing, let's say, played out in the ecological theater? And if this is the case, then we should find positive among individual correlations between these traits. And then finally, the much more interesting question concerning like ecological relationships. Do individuals indeed then occupy individualized landscapes of fear? And do we have to move on from a population level concept to actually resurrect this variation that is in the population? And to make it kind of operational here for my talk, then the prediction would be that among individual differences in perceived predation risk should indeed lead to differences in the spatial temporal distribution of individuals, which is then the definition of the landscape of fear. So what I want to do in this talk, so this is a bit of the roadmap, is I brought you a couple of example studies where I want to illustrate certain aspects that link to these questions. The first one is risk-taking brave little creatures of the night. Then the other example is, towards this question, like no variation in risk. Does this lead to no variation in risk-taking? Then let's see whether spiders are also afraid themselves or just we of spiders. Then asking basically the question of the talk, the title of the talk, who takes the risk in the landscape of fear? And then asking how, if they exist, how might they be maintained, and how and what kind of consequences can they have? So since I've worked myself for 10 years in Madagascar with wonderful nocturnal animals during my PhD in Göttingen and later during a postdoc, I always say there's no good talk without mouse lemurs. So I always have to slip them in, and here they are. We studied them in Kirindi Forest in Western Madagascar, and this is also where I started basically my first work on animal personality with the gray mouse lemur. So that's the one here on the bottom. Exposing them to standardized tests for behavior, as many people did at that time. So I constructed a little wide open box, put it out in the forest, caught my mouse lemur, put it in that box, and filmed it in the pitch dark forest. So no light for me. I couldn't basically see what the animals were doing. So to have basically the conditions that they are used to. And if we put these animals in these boxes and filmed them, then you would see that the animal in the left test would kind of readily go to, for example, this novel object as a little toy car. Whereas if you paid attention to the one on the right side, maybe some people watched that one and thought like, hey, the video isn't working. No, the video was working. And this animal actually only turned its head by, 30, 45 degrees in the whole time it was filmed. And if you do this test again, obviously, then you will see that the one on the left would always respond quickly to this novel object, and the one on the right side would always respond relatively late or not at all. And this is nothing super novel or exciting that many people have done it. What was exciting at that time still was that I could do this over a couple of years with the same individuals and could show that these animals did indeed show consistent individual differences in boldness and exploration even over years of their life. And we could show it with a good sample size for these type of animals. But so far so good. You put animals in boxes, you expose them to super scary little Malagasy toy cars, but what does it actually mean? And to test this, so we did a very simple experiment at that time. We followed mouse lemurs, free-ranging mouse lemurs in the forest, and we put out feeding platforms where we attracted them to come. And these feeding platforms were either, in the normal foraging height of these animals, so in the trees, or they were on the ground. And going to the ground is pretty risky for these animals because there are a lot of predators, and they can actually hear them moving in this dry leaf litter. So these animals are pretty wary before they go to the ground, and they would only go there quickly, for example, to hunt an insect and jump up in the trees again, where they obviously perceive it to be safe. So we use the fact that they don't want to go to the ground. We lured them there with nice prey. and basically did focal observations of these feeding platforms to measure the time it takes them to go on these high-risk ones. And just excuse me for this very small sample size. It's really just an illustration of the principle. And what we saw is that the latency to make contact to this super scary toy car in the behavior test very nicely predicted the latency to enter a high-risk feeding platform of a free-ranging individual in the forest. So with this, we kind of had an ecological validation of our boldness measure, so this animal personality dimension, and risk-taking during foraging in these mouse lemurs. And so far to the brave little creatures of the night, let's go to some more like very controlled experiments that we did further on with other organisms. And when I moved labs, I also moved organisms and actually selected to work for a couple of years and still do with a small rodent, or with different small rodent species, but this one also in particular, that is the bank wall. It's a European small rodent of like, between 12 and 40 gram. It exists virtually everywhere, in every hedge in the forest, along field margins, etc. And what is so interesting about it for my types of question is that it really is a prey par excellence. It's basically, again, like the mouse lemurs, eaten by everything that's there and that's bigger. So kestrels, owls, snakes, if they are there, carnivores like weasels, etc. So a nice study system to use when you're interested in predation risk effects. And what makes it even, comparably nice to the mouse lemurs, it's much easier to catch them in the wild and transfer them to the laboratory for a couple of weeks to do some controlled tests there. And that's what we did. So we measured again their personality using very similar tests to the mouse lemurs. So this kind of open field test. And this test starts with kind of an emergence test where we put animals in a dark tube and just let them basically on their own go into this open field arena and just measure the time, this latency to do this. And this latency to emerge is really highly repeatable over time, independent who does the test. So it's a really nice measure also to do with students in various projects, because it's really has no observer effects, and it's super repeatable, like 0.7 over time, even also again over long term of these animals' lives. And it's a very easy measure to get. And this measure, we showed in many different studies, is very good predictor for what these animals also do in their daily life. And here I used it to explain variation, again, of foraging of these animals. And I set up like a super artificial experiment in the lab, where we had these animals in large arenas of 4.5 by 4.5 meters, so nearly a whole room. And in these arenas, we had a setup of 25 by 25 of these food trays or food patches, which were basically just little white bowls with sand and very few, as you see, like 50 little grains of millet mixed into the sand. And then we had two treatment levels. One was the arena covered, so we wanted to have a more safer condition, and one where the arena was completely open. And then we had also day and night conditions, because these animals usually perceive higher risk when the light is on, when it's basically day in their normal landscape. And you see, I mean, in German, we call this kind of Joghurtbecherforschung. So you use these kind of little white plastic trays that don't cost any money. That's really the lowest tech kind of research you can do. And put the camera on the ceiling and just basically film what these animals are doing and sieve your sand. And sieving sand is really, I really mean sieving sand. So we basically sieved 7.5 tons of sand in this experiment, and I think we counted about 1 million millets in and out. So basically me, with the help of 2 student helpers, were quite busy for a couple of weeks. And what did we get? We get a couple of measures of giving up density. And what we used was the minimal giving up density across these 25 seed trays as our measure of perceived predation risk. We used the mean giving up density across these 25 as kind of a measure for the resource gain, so what the animals actually took out. And we also calculated the coefficient of variation across these 25 seed trays as kind of a measure of the heterogeneity of food distribution that these animals create in the landscape. And this becomes more interesting later on at the very end of the talk again. And what did we see? I mean, we first of all saw that all of these measures were repeatable over time and across treatments. So individuals that had lower giving up density, they also had it every single day of the experiment, and they also had it across different treatments. And I have to say that these different treatments were run over a couple of months. So it was, again, also some kind of measure of longer term repeatability. Then, much more interesting for us than just this repeatability was that actually, again, the boldness that we measured in these behavior tests did predict the minimum giving up density. So the bolder an individual was, the longer it foraged in a patch and took out more seeds. So basically reduced the density in this patch. And if we take Joel Brown's assumption by word, then this should give us a measure of perceived predation risk. So indeed, bolder individuals perceived lower risk in this landscape. And the other side of the coin is if they stay longer in these patches, and they did so also on average, then of course they take more food out. So, you know, they harvest more in this landscape and basically should have a fitness benefit from doing so. And the other thing that was interesting is that the bolder an individual was, it created more heterogeneity in the landscape. So it didn't just exploit every patch to low densities. No, they actually were pretty sloppy in how they exploited this landscape and created a lot of heterogeneity. So if you summarize this or depict it a little bit back to this giving up density graph, then we see that kind of the shy individuals has a higher giving up density because they perceive higher risk. The bold individuals have lower giving up density because they perceive less risk. But also the shy ones are more homogeneous in how they exploit the whole landscape. So if you want for a given, time that they want to invest in this landscape, they do distribute it in a more optimal way if you want. Whereas the bold individuals, they were kind of harvesting in one patch to very low densities, and then the other one they barely touched. So they were, let's say, a little bit more sloppy across the landscape, and in my perception, foraged less optimally across this landscape. And this is also something we should discuss a bit more and think about a bit more. I will not touch it again in this talk. But, optimal foraging theory, we all know that it only works under very strict assumptions. And that's what we teach our students every year. But a lot of variation that people may be found between individuals is maybe due to individuals actually being less or more optimal in what they're actually doing, given their animal personality makeup. So that's what we do in a very, very controlled experiment in the lab. And simply because I moved labs and I actually started to teach at the university where people were working with spiders, namely these wonderful jumping spiders. Philipp Steinhauer was a PhD student at that time, and we just discussed about this project, and then we kind of thought like, why not do something very, very similar with these jumping spiders and actually show that there's some generality in what we actually found with the roles. And this Marpissa muscosa is one of the largest jumping spiders that we have in Germany. And the cool thing about these jumping spiders is that they are predators, but they are prey themselves. So they are preyed upon by, for example, shrews. And obviously they prey on other insects, on insects that they hunt by just jumping and catching them, and then this of course should again have consequences on the primary producers that these insects then again eat. And what I used, a whole big lab for the vole experiments. I mean, just remember 4x4 meters, the arenas, and of course you do several replicated arenas at the same time. You need these keeping rooms, etc. We could do in a on one table more or less with the jumping spiders. So we had a whole room full of jumping spiders that he used for his other experiments too. And then, just designed an open field arena for them, had the dark light emergence test here in the syringe, and then measured the movements and whereabouts of these spiders using atovision in that experiment. And you see kind of some spiders, like the one in the depicted in A, basically didn't move during the whole test, whereas the one in D, for example, was kind of moving all over the place, and the one in B was kind of only more or less going along the edges of the open field arena. So we see, very similar to the voles and the mammals that I work with, usually a nice variation among individuals in boldness and activity that is repeatable over time. And then we created landscapes of fear, and obviously we couldn't use giving up density with these spiders because they wouldn't dig for us. So we designed little compartments of plexiglasses where we put live Drosophila flies in there that were actually moving in these plexiglass compartments. And so the spiders would kind of try to attack them. And we basically measured the time until they gave up to attack these insects in these compartments. Here in the arena marked with 5, you actually see the spider, so you also get a bit of an idea of the dimensions. We had 2 conditions. One was kind of with a brown underground where the spider was really nicely camouflaged, and one that was white where the spider was really exposed. And this very simple setup did the job. And what we could show is that the foraging intensity, so how long they tried to hunt the Drosophilas, differed between the dark background and the white background, so the low and high risk landscape treatment. So that's the proof of the principle that these animals perceive this difference. And as in the other study, we could basically predict the foraging intensity by variation in activity and boldness. I only show you activity here because both of them are highly correlated. So again, bolder, more active spiders would have higher foraging intensity. So in other words, lower giving up density, if you want to make this claim. For the younger career people in the room or individual room, this might just be a nice take-home message. So if you can get access to another study system in the course of your career, try to just transfer your own questions to it and see whether you basically can find it's very comparable results just using a different organism. And if you do that with spiders, you even get the cover of Oikos, as I announced at that time on Twitter, happily. And because I was working with all these very cute mammals like mouse lemurs and voles, and don't ask me, I didn't make it to the cover ever with those yet, but with this wonderful jumping spider we made it, and exactly because it was a jumping spider, said the editor-in-chief at that time. So much about the fear of spiders. How do we do with time? We do fine. So let's actually go to the core question of the talk, and that is who actually takes the risk in a landscape of fear. So we've seen there is among individual variation, but does this among individual variation really translate into differences in landscapes of fear? And for that, we also go back to the small mammals, and we did set up again a controlled experiment, not in the lab at that time, but in so-called, I wouldn't call them semi-natural, they are actually near-natural enclosures, because they are basically natural pieces of meadow that are fenced. You see the fence in the foreground, and this fence is 1 meter deep in the ground, and then about 80 centimeters up above ground. And then these arenas are these enclosures are even netted, so there's no access of aerial predators. There is no access of ground predators because there's an electric fence all around the setup of these enclosures. And if you put a single vole into these enclosures, like these bank voles again, then you can basically confine one single individual in this enclosure. It has the size of roughly 1 home range of a bank vole, a female at least. And you can create your landscape of perceived risk. And that's what we did. We created the vegetation gradient. I hope you see it at this picture, where the vegetation was very super short in the left side of this picture here, where we mowed it to basically 2 centimeters. So this is, soccer field vegetation heights. You can see the vole running around with your bare eye. Then the next zone was 10 centimeters high. So, the vole is kind of in the vegetation, but if you want, you can still see it. Then the next zone is 20 centimeters high, so it should be well protected. And then the last zone was more than 30, so it was on average around 50 centimeters. And we put in behaviorally phenotyped, in that case, only male bank voles that we again tested for activity and boldness in different behavior setups. And in every of these enclosures, we had a set of, again, these seed trays with sand, and in that case, sunflowers mixed in, 3 in each zone. And part of them was monitored using also a PID tag reader, so RFID to read the time that every individual was on certain seed trays foraging. And then, of course, when you want to analyze this data, you have to think about it a little bit, because if you simply take the vegetation gradient as a predictor in your models, it doesn't necessarily give you exactly what you want, because our prediction was that the risk-prone individual would be everywhere in this enclosure, right? So we would get basically data from these individuals foraging in each of these zones. Whereas a risk-averse individual, so a shy individual, would obviously concentrate all his foraging activity to the high vegetation where it's safer, and that's exactly what we predicted, of course, but it will not even give us any data for the shorter fields of this enclosure. So to analyze basically our data, I mean-centered the vegetation gradient for every individual. So I had one predictor, which was the mean vegetation height in a microhabitat where a certain individual foraged. And the second predictor in the model was the deviation of vegetation height to this mean. So basically asking, what does an animal do if it goes somewhere more risky or less risky compared to its average microhabitat? It's called basically mean centering, right? In these statistical models, it's often used in behavior reaction norm approaches. And the outcome was then first kind of our proof of principle. We could see a difference between night and day in giving up densities. Giving up densities were lower during the night, so the animals felt more safe, let's say, during the night than during the day. And this is a nice, obviously, test of, experiment works. Then we didn't see any support for a so-called between individual effect or between subject effect. And this is this predictor of mean microhabitat use. So that's on the x-axis here. And on the y-axis, you have the giving up density again. So no effect of this microhabitat use on the giving up density. And if you think about it for a while, then you will come probably to the same conclusion than I did. This means that independent of where an animal concentrated its foraging, it perceived the same risk. For some, this is low vegetation, and for others, this is high vegetation. But where they concentrate their whole activity, they perceive it as equally risky, and that's given by this kind of no effect in the giving up density. And then the second result was that we found a strong support for a within subject effect, so for plasticity. And this means that if an individual goes to something shorter than usual, then it increases its giving up density. And if it goes to something higher than usual, higher vegetation than usual, then it decreases its giving up density. So it's doing what it's supposed to do. It's showing us that there is a variation of perceived risk in this whole setup. And something I haven't shown you here is that we also showed in these models that individuals differed in their reaction norm slopes. So how steep these slopes of giving up density to vegetation height changes were. So far, so good. And now we thought like, well, I mean, this variation slopes now should be predicted by their personality, right? This wasn't the case in this treatment. So independent of their boldness or activity, the slopes were the same. This was a little bit frustrating and puzzling at the same time. And luckily, I did this experiment with a master student, Pauline Lange, who's also co-author of this paper, who was, let's say, a little bit stubborn. And Pauline really wanted to do this experiment with 2 different treatments. And I said, hey, Pauline, this is already a very complex experiment. It will take you so much time to actually run it with this one single treatment. We shouldn't do this. No, I really want to do this. And what she really wanted to do is to have not only this uniform food distribution that I showed you the results for right now. So that's on the left side, where basically every food tray in the whole enclosure was the same in terms of density that we put in. It was only the risk that varied, but not the food quality, let's say, or the food, the amount of food in the patch. The experiment or the other treatment level that Pauline really wanted to do was this kind of counter risk food gradient, so that you basically have more food, higher densities where it is more risky. So here depicted by larger circles. So we have low density when it is safe and high density when it is unsafe to start with. And we did that. When we do this and link basically now our personality with the giving up density change with a given vegetation change, then we do see indeed what I would have predicted in the first place, that animals that are less bold, that are shy, that are less active, respond much stronger. So for them, a change in vegetation height of 5 centimeters means much more decrease in or increase in risk. So that was a super exciting finding, and we could also show, I only have this here in this little sketch graph, that the slopes of these individuals across the different treatment levels, and we had quite a few individuals for which we had both, were actually correlated with each other. So those, how strong they responded was kind of even repeatable across different treatments. Okay, so far so good. Let us summarize up till here. I think what I hope I could show you, and we did a couple of experiments more that I spare you, is that individuals adjust their foraging to perceived predation risk. And this affects remaining resource landscapes. This goes back here to this variation in coefficient of variation in the highly controlled experiment. Then perceived predation risk and resource exploitation vary among individuals. And with the last experiment and also the others, I think we could show that foraging and landscapes of risk and boldness were functionally integrated. And altogether, to me, basically is evidence that actually the landscape of fear is not a population level concept. It's actually that the landscape of fear should have individual layers, to speak about this kind of GIS landscape analysis language. Again, I mean, so far so good. Nice academic exercise. We could tell it to our ecologist friends and say, hey, you should think about this individual variation in landscapes of fear. Being a behavior ecologist by training, you always are like, yeah, but so what? What does it mean? Where does it come from? And does it have any consequences that are worth pondering about? And this is what we do in a couple of experiments right now as we speak also. And I just want to give you a bit of teasers, how, what we do and why we do it the way, and what we find there in the last, let's say 5 to 10 minutes of the talk. So in an experiment that we just finished in the last seasons, is that we thought of, if you look at a meadow, like this classical place where our study animals live, then the vegetation varies quite a bit. You can even see it on this photo, But there are also some really drastic changes to this vegetation, and this is when we humans come in and basically do experiments on this meadow every year, twice even. So really change this landscape of fear drastically. And what we really wanted to know is, how are these landscapes of fear that might be dynamic even actually maintained? So why do we see that certain individuals are found in certain places and others in others? And to do this, we already had some data from different studies where we also did automated radio telemetry, on this striped field mouse here. And what we saw in these studies and in a couple of these studies is that actually also in the field, I mean, not just in these experimental setups, we do see that boldness measured in these behavior tests actually predict where animals live. So predict the microhabitat we find them in. So this was here a graph for striped field mice. We have them for bank walls. And Philippa, my current PhD student, who's very close to finishing now, she studied an experiment with common walls now, where we actually really tried to manipulate the situation and wanted to see whether individuals really choose to live where we find them actually. And we did this being, motivated in our thinking by a recent conceptual framework that has been put forward by a couple of people at the University of Bielefeld and Münster, also heavily inspired by work of Pim Edelhaar, for example, where people think a lot of, how does this kind of match between an individual and its environment is mediated? What are the processes leading to it? And if you think of an individual, being born in a certain environment, and its phenotype, its behavior phenotype, for example, doesn't match to this environment, then this individual could, simply go and disperse and find an environment where it fits better. And people in this conceptual framework would call this niche choice or matching habitat choice in the theories put forward by Pim Edelhaar and Dan Bolnick and others. Another way to do this is kind of you can actually change yourself. So just become less risk sensitive and stay put where you are, change your own phenotype. And by changing your phenotype, for example, by plasticity, become better fitted to your environment. And that's what these people call niche conformance. And then there's obviously a very large field of animals changing the environment to fitting better to themselves. So a niche construction. And using this framework and also being inspired by the NC-Cube research initiative of Bielefeld and Münster, Philippa and we started to test at least niche choice and niche conformance in an experiment. And what we saw is for these bank walls, and I only give you teasers here because the study is still under review, and maybe one of you is a reviewer and hopefully is positive with us. What we saw also for animals in the field is that the bolder an individual was, the lower was the vegetation we found it. So bolder individuals indeed occupy riskier microhabitats in the field. This, resembles what we found before in other organisms. But now we were pretty nasty, and we used mowers and changed this landscape drastically in situ. So our animals were there while we were manipulating the landscape by mowing a checkerboard pattern into the vegetation. Then caught these animals again. And measured their behavior and asked where did they go to, where do we find them again. And I don't want to give all the original results away here, but just the bottom line is that our animals, those that were risk averse, would basically move away if their home range is affected by mowing and move into larger vegetation again, so that we find them in vegetation height that is basically resembling what we found them before in. So, and this is kind of an experimental indication for niche choice. So animals basically, according to their behavior phenotype, choose basically the risk landscape part that matches their behavior phenotype best. And as I said a few times before, this matches very well to this whole wonderful framework by Pim Hedler and Dan Bolnick, who really ask, how can fitness change? And one way to change your own fitness is by selecting an environment that fits to yourself. And if we think about this, further on, and I think we hopefully think about it also together in the future, then one could basically think about this kind of whole landscape of fear also as part of this extended phenotype framework, if you want. And finally, let's talk also about one or two consequences for species interaction, or at least again give you a teaser on how this might look like. And for that, the question is, how does this kind of variation in forager behavior translates and trickles down to the next trophic cascade, which are the seeds here in this depiction. So do we actually see behavior-mediated indirect predation effects, and can we actually see individual variation in that? And to get this kind of operational and functional, Jana Eckert and I developed kind of a new, let's say, concept and method at the same time, which is not using just only these giving up densities, which were basically one seed type in a depletable patch, but actually think of whole communities of seed types or any other food in depletable patches. And if you do this, so you create a whole, diverse setup of resource species experimentally, and for small mammals, this is really easy. Then you can actually measure the effect of variation in consumer behavior, which obviously is mediated via the predation effect. So the more riskier this consumer feels, the more it changes its foraging behavior, and this foraging behavior variation then has effects on the diversity across whole landscapes. And with using this kind of or measure of cascading effects of variation in foraging behavior, you can basically quantify these effects on the abundance, diversity, and functional trait composition of the whole resource community at local levels, so really at the patch level, but also across whole landscapes. And we put it out in Ecology Letters 2 years ago, and I think it's a really nice measure. If you work with small mammals, try it out. We did, obviously, to have a bit of a proof of principle study done in this wonderful landscape again, and put out arrays of food patches in seven are basically arrays, and we could measure alpha diversity at the patch level. We could measure regional diversity, so gamma diversity across the whole landscape, and we could measure so-called beta diversity, which is the variation across landscapes. And then we basically put that out, put a few camera traps there, and just to illustrate a little bit what kind of different foragers came by. And the PhD student Clara Ferreira was super busy sieving all these patches and counting the seeds. And what we find is what we expect: the higher the vegetation where a patch is, the lower the giving up density. That's what I showed you a couple of times. The higher the vegetation, the lower the alpha diversity, so the lower the biodiversity at this patch. Okay, as predicted. But what is more interesting is the higher the vegetation across the landscape, the lower the diversity across the whole landscape. So if foragers basically feel safe across the whole landscape, they drastically change the biodiversity at this landscape level, which should then have an effect, for example, on the seed banks and the soils, etc. But on the other side, also, the higher the vegetation, the safer they feel, the more variation they create. So you get a very heterogeneous landscape. And this was really just to illustrate you that this might be a nice handle and tool to look into these consequences of individualized landscapes of fear. And we do this also with individual-based modeling that I will not talk about, but so much to the empirical tool. Okay, it's time to wrap up, I think. So I hope I could kind of convince you that the landscape of fear has individual layers. I think we are quite convinced now that boldness and perceived predation risk are functionally integrated. So basically, this is our ecological validation also for this important personality dimension. What I think is that under competition for predation risk-free space, so one of the ecological niche dimensions among individual differences in perceived safety could really act as a form of intraspecific niche specialization or individualized niches. And this is something that excites ecologists at the moment a lot. Then I think that there are also quite interesting links to this idea of extended phenotypes, and so links more into this evolutionary background and explaining this maintenance of variation in risk sensitivity. And finally, I think, yeah, that we are just at the beginning. So, I mean, we showed it now, it's there, but to really get and dig deeper into the consequences of this individual variation, do we really have to plug it into our ecological models of species interactions, and also thinking about how it might affect sociality? So often animals actually live together to avoid the risk of being eaten. But if individuals actually perceive that risk very differently, this might also, get us another handle and spin to understand actually group dynamics or also flock dynamics, etc. And another interesting line of research, obviously, is to understand the mechanisms or processes that actually maintain individual variation in risk taking and perceived landscapes of fear. And with this, I want to thank a lot of people. That was still a lot of work that I did while I was in Potsdam, working together with Jana Eckert a lot, and wonderful students and PhD students there. The first mostly more work was still in Madagascar, also funded by the Primate Center partly, and the spider work was done with the spider gang in Greifswald, Philipp Steinhoff and some others there. And thanks for your attention. I hope it wasn't too long. And obviously, I'm happy to take some questions.
Thank you, Melanie, for such a wonderful talk. We will now take questions. So what I will ask is that if you have a question, please just type a question mark on the chat function, and then I will call you up as they come in. And when I do, just introduce yourself and tell us where you're from, because we are still building quite a community of researchers. So it's always nice to know where everyone is coming from. And if there are also questions on YouTube, I will ask, I don't know, who can check that for us. Yeah. Okay, so let's see.
Yes, Melanie, thank you for a great talk. I really love how you put theory, ecological theory, together with your experiments in the different study organisms. I think it's also a very good way for students to see how to structure your research. I have a question to you. Last part of your study on the matching between the behavioral phenotype and the environment. So it's easy to understand how shy individuals end up in the environment with the lower risk, with a higher vegetation. But what is the cause for the bold individuals to end up with a higher risk? Because for a bold individual to be bold under low risk would still be better, more beneficial than being bold under high risk. Do you think this is secondary because many shy individuals go and then there's more resources for them left in the more risky areas?
Yeah, it's a good question. And it's also actually always the first question that comes, because obviously, I mean, why should you live in a crappy place if you're bold? And one thing you need to know is that from the telemetry studies that we did with several of the small mammal species, is that what we could show is that bold individuals actually occupy larger home ranges, and that they are also better in defending them in a way, so they have less competitors overlapping with their home ranges. And what I think how it works is that basically these safer areas are pretty crowded because all the shy guys go in these safer areas. So the densities get higher. the resources per capita get lower, and the bold ones basically their behavior phenotype allows them to live in areas that are a little bit more risky, but then they have more resources to themselves. So this is the idea that we have, but obviously we have to show this. And so far we are still scratching our head a little bit how we can show this, because measuring resource availability for these animals in the field is pretty hard. So it's impossible, basically. So
But
we need to do this in manipulative experiments.
Yeah, you could experimentally remove the shy individuals and then expect the bold ones to come in. And
Yeah, we did this in a way. I mean, we did set up experiments for a different question where we had different competitive environments. So we had in that sense, the experiment was actually the interaction between two species, so a bit of a different question. But we set up one species, one interacting species as only shy or only bold. And then you would see kind of how these games play out. But these are very big experiments, so you have to do them on the experiment on the population level, and you have to replicate it well. So that here we are not talking about these small enclosures of 15 by 15 meters. We're talking about 50 by 50 meters enclosures and several of them. And so these are quite laborious experiments, but these are the ones to do in the next steps to actually show how this whole mechanism works, obviously.
Okay, thank you.
Thanks, Carsten. The next we'll have a question by Marion.
Yeah, my name is Maren Hock. I'm from the University of Derby. Thanks very much for the great talk, Melanie. I had a thought, and I haven't thought it through, and I haven't thought about it before, but it's very related to Carsten's question, actually. Did you consider looking at the metabolism of the individual? So, because obviously in the dinner principle, well, you don't want to be eaten, but you also don't want to starve. So could it be that the ones that are bolder also are the ones that just need more food and therefore have to be bolder?
Yeah, this is I mean, one of the hypotheses that is out, right, to explain variation in more risk-taking behavior. So in this personality world, this idea that this comes along with kind of higher metabolism. And for our animals, we did actually one experiment because we have this problem that basically in the experimental situation, everyone who's shy gets less resources. So over the long run, they would even lose more body weight than those that are foraging under risk, basically. And they really try to avoid taking risk by all means. So we once set up an experiment where an animal could forage in a larger enclosure. One side was safe, one side was less safe. And there was just one food patch on each side. And they would go to the safe one, until there was basically no single grain in this one anymore. So they would dig through all the sand, kilos of sand to get the last grain, spend all their foraging time there, although the reward was really low at the end, to avoid going to the risky one. So this tendency is really high, and they do pay the costs in terms of staying very long and working very hard for much lower resources. So the question was also, how can they set this off also metabolically? And we did one experiment where we had respirometry running in the nesting chambers while they were resting. And had again these foraging trays out in open arenas. And what you saw is that these individuals that avoid going to forage, they actually lower their metabolism during the resting time. And remember, these are not animals that are known for heterothermy like the mouse lemurs, right? So they are not doing torpor normally, they just lower like 1 or 2 degrees body temperature. But with this, they can already save, this little bit of energy that they might not get when the world is too risky. Coming back to your question, more to this link between the two traits, basically on the population level, there is good evidence for the bank walls that behavior, risk-taking, and metabolism are functionally linked, because people in Poland developed selection lines for metabolism and dragged along risk-taking and aggressiveness. So by selecting for high and low metabolism, you created animals that were also more risk-taking and more aggressive. So these traits are linked in many species, and this might be a nice functional explanation that you take more risk because you have to. But in our experiments, we also showed that they perceive less.
Thanks.
We have a question from Virginia.
Yes, hello. Hello, Melanie. I am Jenny Anatolia. I work in Rome on cat and dogs, domestic cat and dogs, sexual strategy and social behavior. And Melanie, thank you for the great talk. I was just wondering, have you tried to, maybe I missed it, have you tried to diversify the source of risk, like, for example, with odor, like predators' odors or this kind of things? Was just, thinking
Yeah.
about.
Yeah, no, very good question also. I mean, at the moment, I mainly varied the risk spatially. So by, cover, no cover, and temporally only by, light on, light off, something like this. There are really nice ecological theories, also behavior ecology theories, risk allocation hypothesis that also obviously the temporal variation should matter a lot. And for this, it would be nice to have other cues. You cannot remove cover up and down,
Yeah.
every half hour or something. So you want something that you can manipulate more short term. We are a bit reluctant to work with odors because, then you need to have animals in chambers where you can really control the airflow completely. I work in big rooms, to have this in a whole room, remove this predator odor again completely is also not so easy.
Yes.
That's why we thought more about light reflections, or what we tried is kind of predator simulation, so flying silhouettes. So you basically
Yes.
have a flying silhouette of a flying predator. That works very well. And odor only works if the predator actually has eaten the prey organism. So they only react to weasel feces, for example, if these weasels really have eaten bank walls before. But then they also know that this cue, if there is a predator fecal mark, for example, they also know this predator has been there, but it doesn't have to be there at the moment anymore, So they do react to it, but not so much as we thought they would do it kind of on a shorter term basis, basically, to manipulate temporal risk distribution. These odor experiments have been done, many years ago in the labs of Hannu Ylön in Finland with Jana Eckert as well. So where they tested these odor effects and they do work, but yeah, it's not so super practical to use them for some temporal variation in risk. There is some other cool stuff that these Finnish people showed us that the animals seem to emit something like fear pheromones. I mean, really pheromones in quotation marks. So there seems to be an olfactory signal that the animal emits that other conspecifics can perceive that this animal now, was under danger. And this might be something also to elaborate on and use this in further research.
And it doesn't change if they are social or solitary species. I mean, the rodents doesn't change. Emit. Okay, that's
I mean,
interesting.
they did this experiments on bank walls, which are solitary, right? So solitary foragers, females are territorial, males are roaming. So we don't know for the species that are more social or more gregarious, like the common voles, whether they emit the same.
Well, if a solitary species does, then I think that there are more reasons to do if you are social.
Absolutely. So should test this.
Yes. Thank you.
Thanks for the question.
Thank you, Eugenia. So next we'll have Adriana.
Hi, Melanie. Thank you very much for a great talk. And I am Adriana Maldonado Chaparro. I'm professor at the Universidad de Rosario in Colombia. And my question is related to ontogeny. And I hope I didn't miss it from the talk, but I'm wondering if, because we've seen in other personality traits that when you're young, you have one sort of personality, like more bold indeed. And then as you age, you start becoming more shy. And I wonder how this plays out in your system regarding the niche construction, like the individual niche. So maybe younger individuals, they stay in more risk-prone areas. And then as you age, you move more towards like less or low-risk areas. I don't know. Have you looked into this?
I have time for a longer answer again. I have something to say to this. I mean, first of all, we do a study at the moment where we really look at risk-taking relation to dispersal propensity. So we really want to know whether actually dispersal is linked to it. That is actually the core of the PhD of Philippa that I mentioned before. We didn't look at it over ontogeny, but more related to dispersal itself. Concerning ontogeny or the change of your behavior phenotype over life, I actually had the complete opposite result for the mouse lemurs, which I studied over a couple of years. And there I could actually show that males in that case were very shy when they were young and then changed to become bolder when they got older. And there's a really nice kind of adaptive explanation for this. And that is that males that are young, they are still smaller and less competitive in the competition for females. So they might actually protect their future assets by behaving, risk averse. And then they grow, further, become more competitive. And once they are competitive, they basically turn into becoming more bold. And then they again stay, you measure high repeatability at every stage of life, but there is this kind of change over these couple of years where they still grow. This was actually the first personality study that I ever did was on the system trying to explain this pattern, and in that study it was exactly the opposite. So if you have longer living species, then they might actually behave risk averse as long as they are not yet competitive enough and protect their survival, and then turn into, a behavior phenotype that can harvest the benefits of being competitive later in life. Which system did you observe, where you have this opposite pattern? That they when they are young. I mean, humans, yes, of course.
Yellow belly marmots. The young individuals tend to be bolder.
Yeah.
Cool. Thank you.
Thanks, Adriana. So I will take an opportunity to ask a question. So I know with this experiments that you were doing on the different, like the gradient of the food that you give depending on the landscape and the height of the vegetation, did you see if there was an effect of the population size? Because you didn't mention that they're solitary living, but if when the population density was much higher, individuals then tend to shift the giving up densities based on whether the population is high or low and and the direction of the shift, if it's possible. If my question makes sense.
Yeah, it does make sense a lot, because the whole giving up density idea is all about the marginal value of the resource. And if this changes, for example, because food competition gets higher, then it should also change the giving up density. In the experiments that I showed, animals were alone in these enclosures for that reason, to kind of really control for any effects of competition on the outcome of the experiment, because I really wanted to titrate the landscape of fear effects, basically. We now did run a larger experiment in larger enclosures where we had on the whole enclosure high or low vegetation and differences in population density, also kind of in a crossed treatment. And it's not published yet, but what you see is that the density effects are pretty they are basically not there. So really the risk effect is overriding everything in that system.
Yeah.
So then not much variation with population density.
So I have two more questions relating to the same experiment. They're related, these questions. So there's your enclosure with the increase in vegetation height, and first you had the same dishes with seeds, and later there was more food in the dishes that were with the low vegetation. My first question is, did all the voles, independent of the personality, start exploring the dishes with the lowest risk? And my second question is, I'm surprised that the very shy voles still went to the most risky areas. How did you have to get them used to the experimental setup first, or were they so hungry, or how do you explain that? Because a shy wall with grass 2 centimeters high, I thought it would just not go there at all and explore whether there's food or not.
Most of them didn't go there at all. I mean, if you look at
Okay.
I mean, I can't go back to the graphs now, but the between individual effect, right, wasn't there in both treatments. So, and this is kind of the zero for everyone. And the effect was there for the difference between the patch where you are now to your mean. And a shy wall would, for example, only cover this kind of, let's say that let's have their mean in the 20 centimeter, then go to the 30 and go to the 10, but not to the 2. But then you can already estimate the slope, right, around this individual mean. And this is what was shown. So the shy individuals would indeed not really go to the 2 centimeters, even if these were super rich food patches.
Okay, now I understand. Thank you.
Have any other questions?
There's nothing.
Oh, we have a question from the Lema.
Yeah, hi, Suleima Tang Martinez, University of Missouri, St. Louis. I'm retired. I worked on social behavior in small mammals before my retirement. This is more just random thoughts that came to mind, but since there weren't any more questions that I could see, I thought I'd just throw some things out. And you touched on this briefly at the very end. And thank you, by the way. I thought it was a fascinating talk. But you talked briefly towards the end about future directions and about sociality. And it's one of the things, as I was listening to your talk, that I was thinking about pretty much the whole time is what happens in social species, and questions, for example, about whether having more, let's say, shy individuals in a social group would change the boldness or shyness of the bold individuals in the group. So if you have a high percentage of shy, do the other ones that would normally individually be more bold become shyer, or vice versa? How this all interacts at a population level, or at least a group level. And then the other thought that I had is in terms of animals changing their environments to make them less risky. Of course, flocking or living in groups is often thought of as reducing risk, but I was also thinking about social species that have sentinels that watch out and call out alarms when a predator is seen. And it would seem that would be a really effective way of changing the risky nature of a particular environment. And I wonder if you have any thoughts on this. It sounds as if nobody so far has actually looked at this, but I'm not sure. So just throwing out some ideas.
Thanks for these thoughts, because I think we are really thinking along the same lines, right? I mean, these are the type of thoughts I had too, right? I mean, if you think about just aggregations where, you have kind of safety in numbers happening, then, and if you have individuals that perceive risk higher and lower, then obviously a shy individual should, for example, choose the larger flock just to reduce this kind of, individual risk, because you perceive the whole situation as more risky. Or if you think about social groups, I mean, who is doing the vigilance behavior might not be just totally random or totally condition dependent or whatever. It might also be intrinsic differences explaining the distribution of the roles taken and the behaviors taken or performed. So I think that there's a lot to think about and to do still. We're just writing a grant that might include some of these aspects. To dig a little bit deeper and also think in modeling terms a little bit of, what would you expect also in terms of frequency dependence, also of having different types in groups or populations, etc. So yeah, no, super exciting questions. To my knowledge, not too much traveled path yet.
Yeah, thanks.
Thanks, Amanda. I have a question from Lauren.
Hi, Melanie. Thank you for a really great talk. Those of you who don't know, I'm Lauren Hayes. I'm at the University of Tennessee at Chattanooga, and I study social degus in Chile. And I actually had a question about, and I apologize if I missed this somehow, but in the experiment where you manipulate height of the vegetation, are you considering intermediate height to be an intermediate risk compared to tall versus low? Because in some species, like with the degus, we know that if the grass is just tall enough, they can't see very well. And so if you have total cover, you probably have a shield above you from aerial predators, and you probably can see ground predators. But if it's high enough that the predator can see you, but you can't see them very well, that could alter the level of risk. Have you thought about that issue?
Yeah, absolutely. In one study, we had two measures of vegetation. One was kind of the height, and the other one was cover, indeed. And cover we measured at different heights. And there was a bit of a discrepancy actually between the gradient, like where you found the shy guys. So there seems to be something like this also going on when the vegetation is not just grass, but also bushy and trees, etc. In the experiments that we did, I think the grass was the height was just fine, and we were actually pretty lucky, I think, in this whole gradient that we hit it right. I mean, that the variation in the gradient was just perfectly picked to actually show what we wanted to show, to have this titration point where you see the variation. If we would have picked the grass higher, we probably would not have seen any variation there. So I think in that case, the visibility matters through the grass from the view of the aerial predators. And I think the cue that the animals use is whether the vegetation is actually touching themselves and is touching their back. So once they feel something touching their back, that seems to make the difference, and that's what we also exploited in the lab experiments by having just grids, wire grids. So yeah, good point. It might be also more important for animals that actually have good vision to actually see the aerial hunting predators. The voles, they react to shadows flying over, but whether the silhouette is a certain type of predator, they don't really make a difference. So I think once the predator is that close that they could actually see it, it's done. So then it doesn't make a difference anymore.
Thank you. And I have a follow-up question. In these exclosure studies in the field where you have where you introduce them to the environment, do you know anything about these bank voles, they burrow into underground, or are they building little nests on the surface, or both?
Both, yeah.
And do you have any sense of where burrows were located relative to the feeding patches? Okay.
No, we didn't track them like in that experiments. We didn't do radio tracking to see where the burrows were. I suspect that they were in the lower vegetation, in the higher vegetation. Sorry, but I don't know. In the last experiments that we did, we actually, like new experiments, we actually placed a little hut out as well, so that they basically, we try to make them use this one, because they first have to burrow, to dig a new burrow. But they are pretty fast in this, so I suspect that they were somewhere in the higher vegetation, but I don't know for sure.
Okay. So it may not be relevant, but the reason I asked that is because we know that like giving up densities in the degus is also associated with the distance to the burrow.
To the burrow, yeah.
And that interacts with group size actually in unique ways. So interesting to see that. But if they're all burrowing in the tall vegetation, it probably doesn't matter so much.
Yeah. I mean, yeah, but a good point. I mean, we simply don't know in that experiment.
Well, thank you. I really appreciate the talk. It was really interesting, really exciting stuff.
I don't seem to have any questions, and there are no questions on YouTube either. So I think we can stop the live stream and just have a normal unwatched chat.